get the incorrect results. So you can see the quality of testing was very, very, really, really, really wrong. So this led to the production of what we now call the National Genomic Test Directory, and really about this was integrated into the new NHS Genomic Medicine Service. So this was launched in October last year. We've had a large change in the laboratory and clinical services. We're working towards common national standards, specifications and protocols, so we get testing and variability. So anyone referred from any clinical centre should have the same opportunity for a genetic test. And this is achieved through a single, as I mentioned, national testing directory, and that covers the use of which gene tests and how the methodology to be tested for each clinical indication. So we now have 22 rare disease groups going into whole genome sequencing for standard of care, and we have 3 cancers. So we've had to build quite a new different infrastructure within the NHS to be able to deliver this. We have, here's our genomic medicine service here, and underpinned is the learnings from the 100,000 Genomes project through our GMCs, our genomic medicine services, and our clinical services. We now have a national laboratory network, so we have 7 new laboratory hubs, and there's a hub and spoke model, so they will have their specialist services and different local laboratories to perform the testing, as well as the infrastructure through the genomic hub itself. And we have our national testing strategy, which is the annually refreshed and reviewed through peer review of the national testing directory, and that's how we can keep it up to date with this annual peer review. Obviously the data set is something that's very challenging, and we're continuing the collaboration with Genomics England to be able to deliver the whole genome sequencing and integrate into our clinical interpretation pipeline and also to our clinical variant arc, so we can use the learnings nationally to be able to help and drive forward interpretation. And then of course we need to look at workforce development and use our links with industry and academia to be able to translate this data as we go forward. So as part of the National Testing Directory, we have code R21 that we all love and know well so much, which is the clinical indication is for fetal anomalies with a likely genetic cause. So the current testing that we are setting up is to look at the whole exome sequencing or a large gene panel. So the way that we're setting this up is currently not a clinical service funded in the NHS, so it's something very new that is being implemented. We have had 2 UK-based research studies, Page and Boost, and we're trying to focus and use the expertise and knowledge that has been gained through these projects to set up the clinical service. So the progress to date is we've reviewed all of the 7 genomic laboratory hubs to find out expertise and knowledge on where they stand and how close they are to being able to deliver a high standard expert clinical service. It ended up with a selection of 2 centres, which is Great Ormond Street Hospital, which is part of the London North GMC and GLH, and also the West Midlands, the Birmingham Laboratory, both of which were involved in the PAGE study. There's been a national NHS England guidance documents out for consultation, which closed on Friday, so we'll be able to finalise those. And also what I think is really important is not only did we go out to the genomic contacts and community, we also included the Fetal Genomics Group and the British Maternal and Fetal Medicine Society, because it's about engaging all the clinicians and all the key key stakeholders in this area to make this actually happen. So the idea is we want a national standard of testing. So the home GLH will extract the DNA from the amniocytes. They will perform their QF-PCR as standard of care that happens just now. And if negative, then the DNA will go directly to the testing laboratory. And arrays will be performed by the home GLH in parallel to exome sequencing. So this is to demonstrate the effectiveness and the added benefit of whole exome sequencing compared to what the standard of care currently is. The analysis of the whole exome sequencing data will be through the fetal disorders gene panel, which will be applied. So this is very targeted specifically for genes causing phenotypes which may be present in prenatal period and can be detected on fetal ultrasound and imaging. So it's very important we're not looking at absolutely everything. We are being very targeted at the genes that are being analysed, and there's a nice poster that Genomics England are presenting here about how they created the gene panel and how that is going to be applied. And I think also very importantly, people are worried about what we might find, but categorically we're saying that no additional findings will be analysed or returned to the referring clinician. And that's a very quick snapshot of what is happening in NHS England. I'm happy to take some questions. The question was, what's the target turnaround time for the service? I think initially we'll 3 days. Yeah, Lynn's here for 3 weeks. I think initially it will be a slow ramp up because we want it to be very controlled, and we don't want anyone referring any fetal sample just because they know the service is available. So we've got very tight eligibility criteria, and obviously then we will have our QF-PCR performed before the sample's sent, and then it'll be a 3-week turnaround for the WES result to go back to the referring clinician. Lynn says or less.
It's the maximum. So we do QF-PCR and then we go straight to exome sequencing. Yeah. So what about looking for copy number variants first?
Okay, so it's about moving the standard of care to whole exome sequencing. So we're going to do that in parallel. So the NHS will pay for parallel testing of arrays and for whole exome sequencing to be performed at the same time, and that's to make sure that the patient gets the result in a clinically effective turnaround time, rather than reflex testing where it's going to take much longer to get that information back. Yes, in parallel. Probably just for a limited period of time until we have the evidence that it's clinically safe to move to WES. I would say, so did everybody hear that? The question was, if there's very clear clinical indication it was a single gene disorder, would you test for that first, or would you go for WES? I think until the numbers ramp up and we have a really high throughput service, then yes, we would do single gene disorders. I mean, that just seems more sensible. But I think once the service is up and running in high throughput, you would want to run everything through the same pipeline. Yeah. Yes, they are, and that's why we're going to whole exome sequencing rather than more limited like clinical exome, because we will have that data, and if we, after birth or any particular family events, we can go back to that data, and it's there that we can reanalyze if necessary and appropriate.
Can I just say, we've just got a large moment to evaluate the implementation of in terms of what health professionals think, what patients think, and what the needs are, and compare the different views and approaches across the country.
An uptake as well, I think.
And uptake, yeah. So we've got, so that's, we get quite interesting data.
Next year. Or the year after.
Thank you, Sandy, for your talk. Very informative. Then I would like to ask Bryn to come to the stage. Professor Bryn Levy, he's from the Columbia University Medical Center, and he will talk about his laboratory experiences with hundreds of fetal exomes.
Thanks, Diane. So the first thing that you should know is when Diane asked me to speak, I said, well, we're actually not doing this clinically as a regular service. We are still doing this under a research protocol. But her response is, we can still learn a lot kind of from the challenges that you saw in the research mode. So I'm just kind of sitting how we are starting off. Okay, so this is the kind of overall design of the exome sequencing study that we did at Columbia. And I guess the big difference with many of the other selected studies is that we had all patients with fetal structural anomalies, including those with NTs greater than 3.5. We had informed consent, we did parental bloods, we were doing trios only. We did karyotype and CMA, so that is an integral part of the study. The trio was analyzed by whole exome sequencing, and then clinically significant information was reported back to the patient after confirmation in our clinical CLIA lab. So I guess one of the big differences why it became very easy for us to do this was the concurrent initiative by Columbia University, which they called the Precision Medicine Initiative. And here you can see the different facets that they were focusing on, one of them being maternal and fetal medicine. And part of the initiative was the establishment of the Institute of Genomic Medicine led by David Goldstein, who came from Duke. And the overarching goal of the Institute was to create a cohesive Columbia-wide research and teaching environment for human genetics and genomics. And with David comes stuff that although we have our own like personalized genomic medicine sequencing laboratory, what David brought was far beyond anything that we could do, very high throughput, as you can see, this genomic analysis facility, very sophisticated bioinformatics, and then part of the research paradigm is also then to look at the experimental platform, so you could do really so much under this umbrella. This is working. Okay, and then again, here is the bioinformatics that they had spent a lot of time setting up, looking at the sequencing and genotype data, developing these automated standard genomic pipelines, setting up and understanding the differences when you look at the data and the quality control that you would need to get accurate analysis, and then analysis and application development. So the IgM diagnostic analysis, the goal is evaluate novel bioinformatics approaches in identification of genetic diagnosis and new gene disease associations. Bioinformatics have, like most of us know, various filters to qualify the genotypes. They look at quality control thresholds, only consider protein altering variants that meet certain genetic inheritance modes, absent or low frequency in controlled databases. And the key assumption of this approach is that we are dealing with a completely penetrant genotype, which would explain the majority of the clinical manifestations. And so, I mean, this is not news to this group. Variant filtering is really important because taking 20,000 variants down to one, as in this example, takes a lot of work. And so you're looking at de novo or newly homozygous or newly hemizygous, newly compound heterozygotes. You've got to have good quality control thresholds. You're looking, as I said, only at protein altering variants. And you want to see these virtually either absolute or in very low frequency in control databases. Tier 1, we consider high quality with ultra rare, and then tier 2, previously observed as pathogenic. And this is something that we are looking at with a lot of interest in what they call the kind of hot zone, or those that have bioinformatic signatures. And essentially, these are genes that are ultra conserved and they are really intolerant to change. And so when we see these things under those various kind of definitions, the ones that are previously associated with autoimmune diseases, we're going to report out. But what we're doing for those where we see virtually no change or no presence of variation in the populations, but the actual gene or the protein makes sense potentially, but there's been no evidence in the literature, we kind of put those on our watch lists. And what's actually happened as people like Lynn and colleagues around the world are looking at more and more prenatal phenotypes, these watch lists are actually translating to new phenotypes or newly discovered phenotypes that we're seeing in the prenatal stage. Okay, so again, as a disclaimer, we have an outstanding group of genetic counselors who are trained in the IGM, and they are looking at the raw data after the filtering pipelines, and they kind of gave me their slides to give some of the helpful tidbits. One of the things they look at is variant effect prediction, and I kind of took this straight from them, where they paste in the genomic coordinates for a variant and find out the effect on the protein, the intron exon, where the variant occurs. When you search for a variant, you have to format just so. So in the text box where it says either paste data, you'll paste in the genomic coordinates, editing the variants so that the first and last dashes are replaced by space, and the middle dash before the base pair change will be space period space. So here you can see the example. And then after you enter the genomic coordinates, you expand the identifiers and frequency data line, then check the gene symbol, CCDS, protein, HGVS, and click run. It takes a minute for the site to run, during which time you'll see a little gray box that says Q, and when it's done, that box will be green and says done. So I know for some of you that are in the trenches, that's really useful information. Fortunately, I didn't get to do this because by the time we sit in the results, we've had a lot of work done by our counselors. So you're also going to be considering different transcripts. You're going to look at and sample for the express sequence tags, RevSeq transcripts, CCDS, and this is the consensus CDS project, which is a collaborative effort to identify a core set of human and mouse protein coding regions that are consistently annotated and are very high quality. The long-term goal is to support convergence towards a standard set of gene annotations. LRG, which is the locus reference genomic, is a manually curated record that contains stable and thus unversioned reference sequences designed specifically for reporting sequence variants with clinical implications. And in Nomad, you can see the different transcripts that a particular variant is in. So here is the process that we did or used to recruit patients. We had our subject, a referral. They then met with the genetic counselor. Exome sequencing was performed if the array and karyotypes were normal. Then there was analysis and prioritization of the variants. And then we all met together. Then anything that we just deemed as appropriate for reporting was then Sanger confirmed in our CLIA lab and then returned back to provider and subjects. So subject referral, the genetic counselor assesses the patient eligibility. Genetic counselor determines if other family members should be enrolled. The meeting with the genetic counselor, so this is not inconsequential. It's at least an hour. It's a single visit. What happens at the visit? They review the consent form. They discuss incidental findings. They review the family history, draw blood, or arrange data transfer if there was sequencing or something done at another laboratory. Okay, the pre-results meeting, and again, this is a lot of the hard work which is done by the genetic counselors. They look at the variants, they determine clinical fit of the variants, they assess the genetic model of the variants, and then they're going to use many of the resources that we already know, HGMD, PubMed, ClinVar, Exact, Nomad, etc., etc. They're going to be looking at the prediction models, whether it's intolerant, whether it's causing protein changes, etc. And then the results meeting, and this is what many of us speak about, it's that group of clinicians, scientists, particularly those that have experience in maternal fetal medicine, our obstetricians, our counselors, and our fellows and students, and we all get together. Those potential candidate genes are discussed, and then we all kind of unanimously decide on which one should then be reported back, which then here we go. Which then gets confirmed and then reported back. Oops. Okay, so possible results, obviously no clinically relevant variants are identified. The referring provider will receive notification that research results are negative. Negative results are not clinically confirmed and therefore still considered research and cannot be included in the medical record. Clinically relevant variants are identified. The research team believes the variant is likely to find contributing to the patient's phenotype. And the next step, as I said, will be confirmation. Now, the big differences between clinical WES and research WES. So clinical WES, all findings generated in a clear lab. Research, only findings that the study team deemed clinically relevant are confirmed. Variants of unknown significance and negative WES are reported back when you run a clinical lab. In the research mode, we're only reporting back those that are confirmed. So we're not reporting back the VUSs. Typically, one reanalysis can be requested for clinical WES. And in the research, what we do, we've set up structures where the data is reanalyzed every 6 months. There's a cost to the patient or the insurance in the clinical setting, and obviously it's covered by the study in the research mode. The main goal in clinical is to provide results directly to patients, and the main goal in research is to stimulate, the whole research environment, learn a lot about the technology, its limitations, and ultimately gene discovery. Limitations of WES, non-coding regions of the genome are not covered. Some regions of the exome are not covered well. Some types of genetic changes are not covered. And we know these are triplet repeats, copy number variants, large-scale chromosomal rearrangements, some insertion deletions within genes, and methylation abnormalities. And so just to kind of bring that to a conclusion, we published our first big set in the Lancet, and this is really nicely coordinated with Lynn Chitty's group of the PAGE study. And if you kind of look at that data set together, you have well over 1,000 genomes. And when you kind of compare the data, it's remarkably concordant. This is actually updated from what was published. And in fact, it's outdated as we had our results meeting last week with another 30 or 35 cases, which I didn't manage to kind of include because I needed a lot of the other denominators. But this is the updated result from the publication. Here you can see our diagnostic rate of about 14.1%. 87% or 70% of them are dominant, 87% of which are de novo. The recessives are about a quarter, and then X-linked recessive bringing up the rest. Incidental findings are about 4. So that actually comes from screening. That's even outdated for that. I think we're about 1,300 patients or so, and you can see the attrition that goes down to the time they actually enrolled in the study. And one of the things you'll see is our diagnostic rate has increased over time. And you can say, well, why is that? Well, over possible reasons, the early series was too small. One of the things, I think these things are becoming more and more prominent, is ever-expanding knowledge of the prenatal phenotypes, and that's allowed us to reclassify some variants later on as we get new information. And that comes with new medical literature during the course of the study associated with these candidate genes within human disease. And then follow-up postnatally allows us to identify children who are growing into the phenotype. And I think Lynn's actually demonstrated with Soto syndrome a couple of nice cases where it didn't really look, like a possible candidate in the prenatal stage, but later on the kid actually kind of grew into that phenotype. Clearly, when you break this up between single anomalies and multiple anomalies, you're going to see a much higher hit rate for multiples versus singles. And for single anomalies, specific organ systems likely have high diagnostic yields. So here we can see the skeletal anomalies being quite significantly high, CNS, etc. You also see specific genes involved that are associated with syndromic disorders, like over here. But interestingly, we see quite a large cohort of the rasopathies or Noonan spectrum disorders, and here you can see many of the genes involved there. So one of the things that we did, which I thought was really interesting, we said, well, from the WES data that we found, and I think, Lynn, we even included yours, and it's still held up for what I'm going to show you. This doesn't include your data, but we said, how does this relate to expanded carrier screening? So you all know what expanded carrier screening is, but the important thing is that the majority of conditions on these panels are not associated with structural fetal anomalies. We also said at the same time, what's its relationship to cell-free fetal DNA prenatal multi-gene screening, okay? And this, I think, is only done right now at Baylor, where they are looking at de novo Mendelian disorders with severe health consequences, which are frequently associated with prenatal ultrasound findings. And that includes, I believe, 30 genes with an accumulated prevalence of about 1 in 600. And so we asked the question, have these genomic screening advances gone far enough? And the objective of our kind of little study was to say, in a cohort of fetuses from our study, we sought to determine whether the ability of expanded carrier screening and non-invasive de novo dominance, would identify how well they would do at identifying the types of things that we saw in our cohort. So we looked at all our patients since 2015, and we evaluated whether the causative genomic finding was included. In each of 7 commercially available expanded carrier screening panels, I'll show you ranging anywhere from 141 genes in the panel all the way up to 992 genes. And then just the one commercially available non-invasive cell-free de novo dominant panel, which has 30 genes. So all commercial laboratories use different inclusion and exclusion criteria for genes on their panel, and all panels use NGS technology. So here you can see these are our ultrasound findings and the cases that we had, and we said, well, would they have been picked up? These are the recessive because we're looking at expanded carrier screening. And here you can see in lab 1, they would have picked up 0 of those cases. Oops. Can we go back? I don't know if this is working. Oops. There we go. Okay, lab 2. So as you increase, you can see that they would pick up about 11% of the cases. But importantly, it's not only about number, it's about content. And here you can see lab 3, which had more genes, didn't pick up anything. So that's specifically the content that you choose. Lab 4 had more genes, and they picked up, again, the same number as the 158 panel, but different, again, talking about the importance of content. Lab 5 had over 300 genes, picked up 17%, then increasing all the way up to 992 genes. And I don't believe that lab 7 is still in existence. So commercial expanded carrier screening still misses the majority of conditions found in the structural anomaly cohort. And this is a case for further expanding carrier screening to whole exome level coverage. I think it's going to be a great debate for next year's ISPD if you're looking for topics. Okay, then we said, well, what about de novo dominance? And the genes curated to focus on prenatal ultrasound phenotypes like skeletal dysplasias, Noonan syndrome disorders, craniosynostosis, and syndromic disorders. And here we looked at our cases of our de novo dominance, and we compared it to the Baylor panel, and we can see only about a third, just under a third of them would have been picked up. Again, commercial cell-free screening still missed the majority of conditions found in the structural anomaly cohort. And again, a case for further expanded cell-free DNA screening to larger panels. So if we take the largest panel and we couple it with the de novo dominance and recessives, they would have picked up about 43% of our cases. But as I said, that's really not in existence. And the commonly used panels are around about 400 or less. So that means they would only pick up less than 22.3% of our cases. So what would be the advantages of expanding the molecular screening modalities? Cell-free fetal DNA for de novo variants could be done in first trimester, leading to early diagnoses, discussion of options in pregnancy management planning. Expanded carrier screening could be done even in preconception, allowing more couples the option for IVF with PGD and or earlier diagnostic procedures. Current expanded carrier screening is effective in detecting recessive and X-linked conditions not commonly diagnosed by ultrasound. However, the results show that the majority of both inherited and de novo Mendelian disease leading to fetal structural anomalies is missed by currently available genomic screening protocols. So increasing the number of genes by expanded genomic panels or alternatively using whole exome-based screening has the potential for prevention or earlier detection of fetal anomalies caused by Mendelian disease. So what are the barriers? Obviously scalability, you need a much larger infrastructure to process and curate large number of samples. Costs, what is this cost of screening in relation to the economic benefit? Obviously you need to assess healthcare dollars, it's going to be different from country to country. And curation in the absence of a phenotype subvariants may be classified at the US and not reported to patients, and many screening labs do not currently report variants of unknown significance, simply to decrease confusion and anxiety. So in my last 2 slides, I'm going to put this all together. I really like this because I spent a lot of time with the graphics. If you think about what do we get from all the different things that we do, so karyotyping, don't forget karyotyping, because karyotyping, again, the topic is fetal anomalies in the second trimester, so that's what I'm referring to, will account for about 17% of cases. If we add on, we go deeper, we dive deeper, we do microarray, we're going to add on, okay, so it's additive because most of those anomalies will be picked up by microarray, an additional about 6%. We're going to dig deeper down at the gene level, and by West, at least by our data, we're going to pick up about 40%. And so that leaves a big chunk over here. So I spoke about those bioinformatics signatures, and let's call those plausible. And let's say they're all going to pan out because eventually we're going to discover, you know, those signatures that are intolerant, and we're going to eventually do enough work and collaboration that we'll start understanding the prenatal phenotype. So if we add them on, and that's just something that I was doing exon microarrays, again, potentially adding on maybe 1 or 2%, that still leaves us with about 33%. And so there's still a lot of ground to cover. I can tell you that at Columbia, we've now joined ranks with India's group at Baylor and Nita's group at UNC to do a expanded, we're looking at whole genome assessment. Currently, that will be done, but only the West-based analysis will be reported back. But the nice thing is potentially as we gather that data and follow up these patients, hopefully we'll start to be able to fill in some of that information, potentially from things that we see on whole genome sequencing that we don't see on the West. So unique challenges in fetal West, determining the pathogenicity of any given variant in the genome requires accurate phenotyping as well as bioinformatics resources and expertise. There is incomplete phenotypic fetal data due to the limitations of ultrasound and the temporal limits inherent to fetal development. Determining the pathogenicity of any given variant in the genome requires accurate phenotyping as well as bioinformatics resources and expertise. So case one, just for illustration, was prenatally diagnosed hydrocephalus and aqueductal stenosis. Testing revealed de novo variant in DYNC1H1 associated with dominant intellectual disability with neuronal migration defects. Postnatal imaging did not confirm hydrocephalus, however, it did confirm dysgenesis of the corpus callosum linking this variant to the Provence phenotype. There is a lack of data on prenatal phenotypes in the existing databases, possibly because the most severe phenotypes are embryonic lethal or cause fetal demise and therefore never tested or documented in databases. So in case two, kind of as an illustration, prenatally diagnosed bilateral renaogenesis, anhydramnios, and complex congenital heart defect. Postnatally, the neonate had skin sloughing of the scalp consistent with aplasia cutis and nail dysplasia. A dominant variant in KCTD1 associated with autosomal dominant scalp ear nipple syndrome was found, and this is the first published prenatal presentation of SEN to our knowledge. And these prenatal findings further expand the phenotype of this condition. Finally, there may be differences in the inheritance models between previous described childhood cases and fetal cases. And here, in the third case, prenatally diagnosed pleural effusion, scalp edema, ascites, and IUGR. The pregnancy resulted in a fetal demise. Was found to have, a variant in RYR1 in trans. RYR1 is associated with malignant hypothermia when dominant in central core disease when recessive. And both parents did not have a history of symptoms of malignant hypothermia. So what's the solution? We have to work towards standardizing data collection and collaborative dialogue to further generate prenatal genotype-phenotype correlations. Is certainly required to expand fetal genomics in a meaningful clinical use for our patients and providers. I'm going to stop there. Thanks very much. I do actually need to acknowledge the IGM team, our PGM team, our MFM team. Really, it's a collaborative effort. It's tremendous amount of work that gets done by our counselors in the IGM before we actually have our results meeting. So I do want to acknowledge them, and thanks very much.
Thank you, Bryn, for your talk. Are there still questions, urgent questions? Otherwise, I suggest that we move on to the next speaker. And that is Dr. Natalie Chandler from Great Ormond Street NHS Foundation Trust of the UK, and she will talk about laboratory logistical issues of implementing rapid fetal mass.
Hi, thank you for the opportunity to speak today.
Move back to the other computer. So otherwise Nelly's going to represent my stuff.
So I've been tasked with talking about how we're actually going to implement the NHS England prenatal exome service in our lab and discussing the logistical challenges. Yeah, that would be, thank you.
Just say next slide when you want to.
Next slide. Okay, so this is our experience, or at least my experience working with prenatal exomes. So we've been part of the BOOST project, where we've been doing prenatal exomes in this skeletal dysplasia cohort. As you see, we've had 23 diagnoses out of 29 cases. So we have a very high diagnostic yield. Why is it so high? Well, actually, we've been very much gatekeeping our referrals. We don't just accept every case. We actually look at them and assess whether they're likely to be a genetic cause. And this is really important. So the time to diagnosis is now 10 to 12 calendar days. And we collect all the outcomes for the pregnancies and follow them all the way through. So this shows the real importance of doing prenatal exome because normally skeletal dysplasia is thought mainly to be associated with de novo mutations. Actually, 8 of our cases have been autosomal recessive, so obviously have a higher recurrence risk. We have 13 that were de novo, but more importantly, we've had 2 maternally inherited cases where the mum wasn't thought to have a phenotype, but actually we found this variant and then we went back and looked at the mum. But in both cases, she did have mild symptoms. So you've got the variable expressivity associated with dominant disorders, and some disorders as well can present a lot more severely prenatally. So this is why when we do the NHS England panel, we will be analysing every single variant and not just applying inheritance filtering, because we've got 2 out of 29 cases here. So we think this is very important. We had 6 undiagnosed cases. One of those has now got normal development, so it's probably a true negative. One had a post-mortem after neonatal death, and which showed placental insufficiency was a likely cause, so again, probably a true normal result. There's 2 which do have the skeletal phenotype from birth, but they haven't yet been diagnosed. And then one was stillborn with no post-mortem, so we unfortunately can't follow up that case. The other one is ongoing pregnancy. Next slide. Okay, so we've now been tasked as being one of the 2 testing laboratories in England as part of the NHS England restructure. We have a large number of logistical challenges that we have to address as a testing lab. Primarily is communication, because we're not just working with our local teams, we're working with now half the country. Sharing of images, it's really important we have images available to be able to interpret our variants. Different DNA extraction protocols, so one thing that hasn't been discussed yet at this meeting is, do we have enough DNA to do all of this testing? So we have to have agreed protocols and policies in place between the labs and hubs. Next slide. So what is the testing laboratory's role? So we have 3 weeks. So if we're receiving the DNAs, then we don't have to do the DNA extractions, but also we're doing our local hubs, so we might have to do some extractions. The obvious thing, we have to do the test, so we're doing hopefully trio exome library wherever we can, although if we can't get the trio, we will be accepting duos. Obviously do the sequencing, and then we have our own in-house pipeline that we use to apply, and then apply the fetal anomalies panel. The largest part of our work will be in the variant interpretation, and then probably the most important part will be the NDT discussions, which will be started by email, and if getting complicated, they will then move on to teleconferences. Then we need to decide whether the confirmation test is required. I'll go into this in more depth later. And at the end, we'll issue a report. So this has been discussed earlier, but how is whole exome sequencing going to fit into the prenatal pathway? So all cases will be referred from the FMU into their local clinical genetics team, and they will review the scans together and decide with all the expertise whether they think a genetic cause is likely. As I say, this is really important. You need to gatekeep your referrals to use resources correctly. If it's deemed a monogenic cause is unlikely, they will then undergo standard management. They still may qualify for a microarray. If a likely monogenic cause, we'll have the QF-PCR going on while all of these decisions are being made, all of this MDT referrals coming in. So the QF-PCR will have been done, and so at the same time, we'll be running the whole exome sequencing and the microarray. However, the microarray is not going to necessarily be performed in our testing lab, it might be performed in the local lab, so we need to be communicating at all times. So then I've looked into the sample demographics from the BOOST study, and do we need to culture samples? So through the BOOST study, we had 22 amnios, 6 CVSs and 1 fetal blood referred at a large range of gestations. All the CVSs and fetal bloods obviously weren't cultured, but the amnios, we did actually test 5 culture samples out of 26. Just to go into those in more detail, so 4 of them were from one referral site. Now this site is known to use a DNA extraction method which gives a very low DNA yield. This is why we've had to wait for cultures in these cases. The other one was an internal case, but had array before we did the testing, and therefore there were cultured DNA samples available, so we used these for testing. But I looked back in the original sample, and we would have had enough to do a whole exome sequencing. So how much DNA do we need? So in our local lab, I worked with our CITO team and came up with some numbers. So QF-PCR requires 30 nanograms of DNA. Microarray requires 250 nanograms. Obviously, this is going to depend on which is the testing lab and the method used. And our current whole exome sequencing method uses 200 nanograms. All of these are measured by qubit rather than nanodrop, much more accurate quantification. We are currently validating another method which will take us down to 50 nanograms. So in total, we need 400 nanograms of DNA. So it's not a high DNA yield required to be able to do these tests, but you may also then need a little more for the confirmations. So usually, you shouldn't require culture. But it's important to have this as a backup in case there's technical failures, so it's important to always do this. As I say, we have one centre consistently referring cases for low DNA yields requiring culture. So the issue we know is down to the extraction method by that lab. So we want to introduce standard DNA extraction protocols across the centres to prevent this. But what do we do if DNA yields are too low? Again, this is all down to communications and MDT discussions. The local clinical geneticist will have the experience to know whether whole exome sequencing or microarray is more likely to yield a diagnosis. So they will review and decide which tests they think should do first. Obviously, while this is going on, the sample's being cultured. So they'll do that test. If they identify variants that are thought likely to explain the imaging phenotype, then obviously then we don't need to do the other tests. However, if there's no pathogenic variants identified, you can then move on. Okay, so I think one of the most important stages is going to be the multidisciplinary team discussions after we've got our variants. And this is even important when, as our view of the variants, we don't think there's no causative variant we've identified, because they might have key genes in mind So we'll still report back to them and say, are there any particular genes? And we might have a class 3 sitting there that they then might be able to put the pieces together, and we might be able to get it up to being causative. For every case, we will be issuing a formal report, even if we decide there's no likely causative variants. The MDT input will be even more important with cases where we find variants that we think are likely to be significant. And we will be emailing the details on these variants, including all the evidence we've used for our classification in the email to the clinician, the FMU, the clinical geneticist, and impress feedback. Then we'll decide as a team if they want the results confirming and what could go in the report. So this is one way we're trying to streamline processes in England. So at the moment, we are confirming every single variant we find by next generation sequencing. However, we now have over 7 years of experience with next generation sequencing. So we're doing laboratory audits to set some quality thresholds of when we know a variant is going to be real. So that hopefully should reduce the need to be able to have to confirm every single variant. So therefore, in future, when any variants above our quality threshold, we will not necessarily have to confirm them. However, that has its own limitations because this is a prenatal exome. The family are likely to want prenatal diagnosis in the future. So we need to really make sure we're discussing this at the MDT and say, for speed of result, we're not going to confirm this. However, please let us know if they're considering another pregnancy, and then we might want to go ahead and confirm the variants. We are only going to be confirming clinically actionable variants, so only variants we've agreed at the MDT that are thought likely causal. We're not going to be confirming all these class 3 variants. And we're going to be making it very clear in our reports if we're not confirming the variants. Indels and frame shifts, this is a different because these are often called incorrectly by next generation sequencing pipelines, so these must always be confirmed when reporting to make sure you've interpreted the variant correctly and the variant is what it says it is. But again, we'll only be doing clinically actionable variants. Exome deletions and duplications. So we will be looking at this in the whole exome data, but we're only going to be confirming them again if they meet quality thresholds. We've again gathered a large amount of experience. There's a lot of false positives that come up that we see all the time, so we're not going to spend time confirming those. We're only going to confirm them if they look real. And you can confirm them using microarray. So again, you've got communication here with your testing lab. I've got something, have you found it on microarray? That will save an awful lot of work. So what are we going to report? So we will only be reporting pathogenic or likely pathogenic variants that are related to the phenotype seen on imaging. We're not going to be reporting all the unclassified variants, the class 3 variants of uncertain significance. There will be a few limitations made potentially at MDT discussions. For example, you've got a clearly pathogenic variant in a recessive gene and another variant in trans, which is a class 3, and you just cannot get the evidence, but it highly matches the phenotype on imaging. So there may be the odd exception. Incidental findings, so the use of a panel approach means that these will be limited. It's not what we're aiming for. However, there are genes on the panel, such as the Fanconi anemia genes, which obviously have risk of cancer for the parents. So again, we're not just going to go putting these in a report, they will all be discussed at MDT discussions and will allow then the management of the family. So in summary, introducing whole exome sequencing in the prenatal setting as part of a national study, it has lots of challenges that we're going to be addressing. Having clear policies and protocols in place agreed nationally is hopefully going to reduce these challenges, and multidisciplinary team is going to be essential. Large amounts of communication. This will allow us to then provide an effective service to help the patients. Our data shows that the rapid turnaround time is possible, and these results can then inform on the pregnancy management. Postnatal follow-up in the study we've got funding to do is going to be really important in determining the relationship between the prenatal findings and genotype. So we might get more diagnosis when the baby's actually born from having done this. And thank you. I'd just like to acknowledge the team. There's a lot of people involved in this, so thank you.
Thank you very much, Natalie. Other questions? We have some time. I have a question for Bryn, in fact. Is it possible to ask it? Maybe Natalie, you can also spend there. Maybe there are other questions popping up. You are still doing first a karyotyping in an array. Why are you still doing the karyotyping? Because most of the chromosome aberrations you will also detect with array, isn't it? Well, at least the clinically relevant ones.
So a lot has to do more with where we are in the country and some of the regulations. So we are still in, we're in New York and subject to regulation by the New York State Department of Health. And right now, they still kind of consider karyotyping as a gold standard. So the patients pretty much get everything, you know.
Yeah.
Seems to me doing karyotype is important for de novo apparently balanced transplantations.
Right.
Unless you do long read sequencing.
Right. I guess the argument, again, so I fully agree with you. The counter argument that I've heard is that the majority of balanced translocations are going to be innocuous, you know, and that there is reproductive risks associated with those. But if it was unbalanced, you pick it up by the array. So even if you don't identify the parents as carriers, if standard you know, practice continues for the next pregnancy, an unbalanced form will be picked up. So then you're talking about the rare events when there is actually a phenotype with the translocation being causative by either dissecting, you know, critical region or a gene. And those ones you would miss, but our counter argument is that those are extremely rare.
It would make Cynthia Morton unhappy.
Yeah.
What percentage of cases that are being referred are considered to be likely genetic, monogenic in your case? So what percentage of cases are you accepting to do? I'm asking for that.
We don't reject that many. I'd say probably about 1 in 5 are rejected, but that's just a number off the top of my head. Again, it's just really important. We always make sure we get the images in, and we're lucky to have Lynn, who's been an expert on the skeletal dysplasia, who's able to expertly review them for us. We won't be able to do that when it's half the country, so this is why we're relying on the local clinical genetics teams to do the gatekeeping for us.
And so far, when we've rejected them, I think things like IUGR we've had recently because we've tried to follow them up and they've actually had something else. So they've been correctly rejected.
Other questions?
I will just add, Art, to in your favor. Before Dorothy Warburton, who I worked with for many years at Columbia, she passed away a good number of years, but something we never got to publish was kind of looking, what does karyotype give you that, this is an on-situ question, that you don't get from array. And actually, the unpublished data was around about 1 to almost 2% of things like low-level mosaics, things that you would miss by array, and you'd miss, you know, obviously by sequencing, and then balanced translocations and a couple of other things. And if you actually look at it, it sounds low, but remember how excited we got with microarray when we said it added 1.6% additional information. So it's not inconsequential in terms of what it adds.
What is everybody doing if you find a clear pathogenic variant that has nothing to do with the ultrasound phenotype, like the fetus with Rett syndrome? Like, gets reported or not?
For us, that will come down to the NTT discussions, and we won't be looking for it. It's only if we have our panel, and if it's on the panel, it will come up, but that panel is only there to detect structural anomalies. So yeah, we won't be reporting things unless they fit the scan phenotypes.
I'll check, but I actually think we have reported NECP2 clearly pathogenic. Yeah.
And if you do exons, you should see them.
You had 4 incidental findings. Can you tell something about those cases? What were these?
I don't remember.
Okay. And for my maybe I didn't hear it. Natalie, you are using a gene panel, but Bryn, do you do a whole exon or also a gene
No,
panel?
it's a whole exome.
A whole exon, and then 4 incidental findings on a total of how many pathogenic findings did you have? That was not that much.
The total was, I mean, it was around about 14% of cases,
Mm-hmm. Okay.
the majority being dominant enough.
Yeah. Okay. Well, it's something that we are afraid of, finding incidental findings that do not fit. But is it a real problem?
But so remember, for us, anything that doesn't fit, and unless it's like clearly pathogenic, like art, we're kind of putting on our watch list. And again, we have that luxury because we're in a research environment. So, that's a big difference between when you operate in a clinical lab. And the consent form is also quite lengthy in terms of asking patients what they want, incidental findings. Yeah, those are reported. I think we are including the 59. I have, Nita and Inya, for our protocol now with the whole genome, are we reporting the SMG 59?
It varies by lab.
But for the study protocol? Oh, right, each lab gets to report.
We are, but I don't think
So in most cases, often we translate exactly our protocols. I'm not saying it's going to be like that when we transfer, but we kind of are comfortable with the protocols that we establish in the research paradigm and bring them in. Although the big difference will be including VUSs. That will be the major difference.
in our consent form, you have to agree to the 59 example.
Right. It is an option on the consent form, but it's not done automatically, right? Unless it's checked off.
When you said that you weren't reporting things from whole genome, but suppose you have whole genome and you see an exonic mutation that was missed by the exome. When do you have the whole genome data to realize that? And I assume you would report that?
So I think the protocol's a month and a half, right? Since it was launched. I think the whole genome data is going to be only analyzed at a later date.
So the protocol says that we will analyze and report the digital exome, but then there's going to be research
Yeah, but
to look what else.
I think the problem with that will be we will get to the whole genome data. So your question is, when we get to it and we find something, will we go back and report it?
It will be discussed. There's several committees
Right.
that actually can do say final oversight committee, and this will be the basis going to that committee to make the determination. Okay, there was a question over there.
I just want to mention for example, the one that you presented with RYR1 gene mutation. So what do? Do you run after all the members of the family who are heterozygote and talk to them about malignant hyperthermia? And especially because you probably saw our publication, none of the family members who are carriers develop malignant hyperthermia, and even the one who is compound heterozygote or homozygote had surgeries and did not develop malignant hyperthermia. And the same about the Titan gene.
So this was just from getting a comprehensive family history. You know, interviewing the parents and asking them about that. You know,
So it's interesting because I've been thinking about this. What do we do with those parents? And I actually just last week emailed Bob Dirksen, who wrote the gene reviews on RYR1, and his advice. He made the same point as David that the parents seem to not be at risk, but his advice was still to let the anesthesiologist know just in case. I know Susan Hamilton at Baylor Lab does research on RYR1 and RYR2, and I think they're seeing some difference in the type of mutations that are associated with the. I guess they're beginning to do mouse studies on genotype phenotype correlation. So it may be that these are just different mutation types.
I think we have to swap the questions now because we still have two different things that we have to do. I would like to ask you to stay there and move on with Felix talking about the CMA survey.
so I think we're pressed for time, so I'll be very quick. And what I'm going to announce is an initiative that was undertaken by myself, Diane, Francesca, Elaine, and Sandy. And this is really in response to all of us who look at CMA data, microdata, understanding such significant differences in reporting practices. Sometimes within a colleague of yours in the same lab, and very often labs within the same city and labs across the country, and certainly labs across the world. And so we've been very interested to understand these differences in reporting practices with real pipeline goal of maybe unifying reporting practices ultimately. And so we've created quite a lengthy survey, and this will be published and advertised through as many different channels as we can do, through ISPD, through CAP, through Sandy, and all the professional organizations that we have. So please be on the lookout for this. I really urge all of you to take the time to actually complete this. And particularly those of you who have experience with looking at microarrays, it's going to be looking at reporting practices in prenatal, postnatal, and miscarriages, POCs, really to understand them. And then we're actually looking at specific CMVs that are considered to be kind of controversial. So you can imagine ones that are associated like with autism, whether that's going to be reported prenatally, and people may report some prenatally, and pediatrics are not reported prenatally. So we trying to get an understanding of that. So please be on the lookout for this, and I know it is quite lengthy, but I really would urge you to take the time to complete that. So.
Okay, and then I want to give the word to my co-chair, Elaine, who will tell something about the Global Education and Outreach Committee meeting from yesterday.
Thank you. And thanks to our speakers today. There's a brilliant meeting we've had. Thank you.